Hi, my name's Lexi Jungbluth, and I am here to present case connections for Unit 4. I'm going to talk about urea cycle overview, urea cycle disorders, beta oxidation overview, and beta oxidation disorders, and then concluding with case 1 deductions in part 1. So the urea cycle, that is a removal of ammonia as urea. The first step is condensation of CO2 plus NH4, H4, and ATP to form carbamyl phosphate. This reaction is catalyzed by carbamyl phosphate synthetase 1. It takes place in the mitochondria and requires N-acetylglutamate as an allosteric activator. Step 2, carbamyl phosphate plus ornithine forms citrulline. That's catalyzed by ornithine carbamyl transferase, and citrulline is then shuttled out of the mitochondria. The third step, citrulline plus aspartate to form arganosuccinate, and that's catalyzed by arganosuccinate synthetase. Fourth step, arganosuccinate is cleaved by arganosuccinate lyase to form fumarate and arginine. Arginine then continues through the urea cycle, and it is cleaved to form urea and reforms ornithine. This reaction is catalyzed by arginase, and the ornithine cycles back through the urea cycle. The urea cycle disorders are characterized by hyperammonia, encephalopathy, and respiratory alkalosis. So just to highlight one of these, N-acetylglutamate synthetase deficiency, we know that N-acetylglutamate is required for carbamyl phosphate synthetase 1, so a deficiency in this enzyme would then create deficiency in the urea cycle. Also, citrullinemia, that's a lack of arganosuccinate synthetase activity, and arganosuccinate aciduria would be a metabolic defect in arganosuccinate lyase. So beta oxidation, the oxidation of fatty acids to acetyl-CoA in the mitochondria. Fatty acids are first converted to an active fatty acid, which is acyl-CoA. That reaction is catalyzed by acyl-CoA synthetase. And then the second step, carnitine palmitoyl transferase 1 transfers acyl-CoA across the outer mitochondrial membrane, forming acyl-carnitine and releasing the CoA. Step 3 and 4, carnitine acyl-carnitine translocase binds the acyl-carnitine, transfers it across the inner mitochondrial membrane in exchange for carnitine, and the addition of CoA reforming acyl-CoA, and this is on the inner mitochondria. That reaction is catalyzed by carnitine palmitoyl transferase 2. The second part of beta oxidation, this is a cyclic pathway, and we're breaking down the long-chain fatty acid into acetyl-CoA. This reaction, the first step is the removal of 2 hydrogen atoms forming acyl-CoA, and that's catalyzed by acyl-CoA dehydrogenase and requires FAD. So acyl-CoA will be our final product along with acetyl-CoA until the complete degradation. So we're losing 2 carbons each cycle, and then if it's an odd chain fatty acid, this will be cleaved until the remaining 3 carbon, which is also known as propionyl-CoA residue, remains. And that propionyl-CoA goes on to be converted to succinyl-CoA, which is a TCA substrate. So there are several disorders that can be associated with beta oxidation. A carnitine deficiency, that would be inadequate synthesis of carnitine or a possible leakage of carnitine. And as mentioned, that's required to help bring the fatty acid into the mitochondria. So with case number 1, we're presented with a newborn. She's normal at birth, but within 24 hours she develops lethargy, hypothermia, and apnea. So if we wanted to determine if she had a urea cycle disorder, a urinary acid and a plasma glutamine test would determine that. If we thought it was a beta oxidation disorder, urinary acid, blood glucose, and serum fatty acids would determine that. However, the plasma glutamine test would be best to distinguish between the two, and according to PubMed, an increase in plasma glutamine concentrations are almost always associated with hyperammonia in patients with urea cycle disorder. The test results for newborn female, she had elevated ammonia levels which rapidly increased, elevated urine uric acid levels, and plasma citrulline level was normal. So the elevated ammonia and urine uric acid would point to a disorder in the patient's urea cycle. Uric acid is generated by an increase in carbamoyl phosphate. So, and this is due to a downstream block in the urea cycle, such as a deficiency in ornithine transcarbamylase or arganosuccinate synthetase. Now, since this patient's citrulline levels were normal, we know that the carbamoyl phosphate synthetase 1 enzyme as well as the ornithine carbamyl transferase enzymes were functioning. But the high levels of ammonia and urine uric acid would therefore indicate a disorder with arganosuccinate synthetase. So part 2, I'm going to talk about purine synthesis. I'm going to talk about salvage of purines, purine synthesis and salvage disorders, pyrimidine synthesis and disorders of pyrimidine synthesis, following with the conclusion of case number 3. So purines include adenine and guanine. They have a bicyclic structure, which is more difficult to break down than pyrimidines, which are just a single ring structure. The first step is the transfer of 2 phosphoryl groups from ATP to carbon 1 of ribose 5-phosphate, forming phosphoribosylpyrophosphate, pyrophosphate, and that's catalyzed by PRPP synthetase. And our synthesis continues to add that second ring and forming IMP, or inosine monophosphate. So the salvage of purines. Purines can be recycled using enzymes adenine or hypoxanthine guanine phosphoribosyl transferase, HGPRT. And the recycling of purines reduces the amount of purines degraded, which reduces the amount of uric acid contribution. So there's many disorders of purine salvage, hyperuricemias, which would be elevated uric acid, which would be gout symptoms. Those could include Lesch-Nyhan syndrome or von Gehrig's disease. Hypouricemias, which would be adenosine deaminase or purine nucleoside phosphorylase deficiency, or alcoholism. Pyrimidine synthesis is the synthesis of thiamine, cytosine, and uracil. This is a single ring structure and water soluble. And this reaction, the first step is CO2 plus glutamine with ATP to form carbamyl phosphate, and that's catalyzed by carbamyl phosphate synthase 2. And this reaction takes place in the cytosol, with our final product being UMP, which can then be metabolized to CTP and TMP. So there are several disorders of pyrimidine synthesis, and those are noted here. So in our case number 3, we have a 47-year-old male with severe pain in his right toe, no trauma. We've diagnosed him with gout, and he has elevated urate in his plasma, but decreased urinary uric acid levels. So gout occurs when urate crystals accumulate in the joints, and the body produces uric acid when it breaks down purines. So our patient has elevated plasma urate, which would suggest an overproduction of purines. So the possible deficiency could be Lesch-Nyhan syndrome. However, this is a very rare inherited disease, and symptoms typically present within the first few years, so it's not likely that this is our patient's cause of gout. There could also be a deficiency with glucose 6-phosphatase. However, this is also a rare and inherited disease and not likely our patient's reason for gout. But decreased urinary excretion of uric acid, and according to the Mayo Clinic, you're more likely to develop gout if you have high levels of uric acid in your body. and this patient consumes an excessive amount of soda, and drinking beverages sweetened with fruit, sugar, or fructose does increase your uric acid. So therefore, this patient's gout symptoms are due to an excessive consumption of high fructose corn syrup. Thank you.